The overall goal of this procedure is to generate an in vivo human vascular network. This is accomplished by first culturing human blood-derived endothelial colony-forming cells and human bone marrow-derived mesenchymal stem cells. Both cell types are then mixed into a collagen-based gel, which is injected subcutaneously into an immunodeficient mouse. 7 days later, the implant is harvested and evaluated for the formation of a human vascular network. Ultimately, the presence of human-specific microvessels inside the implants can be verified through histology and immunohistochemistry.
The main advantage of this technology over existing methods, like surgical implantation of construct, is that it's simple and easy to perform, and it doesn't require surgery.
Begin by thawing 1 vial of ECFCs and immediately dilute the cells in a 15 ml conical containing 10 ml of DMEM. Spin down the cell suspension at 1200 RPM for 5 minutes and resuspend the cell pellet in 10 ml of warm EGM2 medium. Then, transfer the cells to a 100 mm tissue culture plate coated by 1% gelatin and incubate the cells overnight in a humidified incubator. The following morning, gently aspirate out the medium and any unbound cells. Then feed the bound cells with 10 ml of fresh EGM2 medium. Continue this process over the next few days until a confluent monolayer has formed. Then wash the cells with 10 ml of PBS, replace the PBS with 2 ml of trypsin EDTA, and return the plate to the incubator for 5 minutes. After 5 minutes, under a microscope, gently tap the plate and check if the cells have detached. If not, repeat the brief incubations with gentle rocking. Once the cells have completely detached, add 8 ml of EGM2 medium to the cells and transfer the cells to a 15 ml conical. Count the cells and spin them down at 1200 RPM for 5 minutes. Now, plate the cells on tissue culture plates coated with 1% gelatin at a seeding density of 5,000 cells per square centimeter in EGM2 medium. Incubate the cells and feed them every 2 to 3 days with EGM2 medium. Repeat this procedure for subsequent passages. Between passages 4 and 8, the ECFCs will be ready to use. This protocol can also be used to grow up MSCs. Just replace EGM2 medium with MSCGM medium, use an uncoated culture plate, begin subculturing at 80% confluence, and subculture at a seeding density of 10,000 cells per square centimeter. This experiment requires 800,000 ECFCs and 1,200,000 MSCs per implant. Generally, 5 implants are prepared simultaneously. To begin, aspirate off the cell medium from the culture plates and wash each plate of cells with 10 ml of PBS. Replace the PBS with 2 ml of trypsin EDTA and incubate the plates with gentle rocking. Check the cells under a microscope every 2 to 5 minutes until the cells have all detached. Then, add 8 ml of DMEM and collect the cell solution in a 15 ml conical. Count the cells and transfer enough cells for 5 implants into a single 50 ml conical. Then spin down the cells at 1200 RPM for 5 minutes and remove the supernatant. Carefully prepare a mixture of fibrinogen and collagen fibronectin on ice. Then, very gently, resuspend the cell pellet in the ice-cold mixture, avoiding the formation of air bubbles. Load the cell suspension into a 1 ml syringe and attach a capped 26 gauge needle. Keep the loaded syringe on ice until the cell mixture is injected. Anesthetize 4 6-week-old athymic nude immunodeficient mice using an isoflurane chamber. Once the mice are anesthetized and unresponsive to toe pinch, proceed with the injections. For each mouse, use a 30-gauge needle to inject 50 microliters of thrombin solution subcutaneously into the upper dorsal region. At the same location, inject 200 microliters of the cell mixture using a 26-gauge needle. The injected macromolecules will gel and form a small but appreciable bump under the skin. After the injection, place the mice on a layer of gauze for comfort and warmth. Observe them until they become ambulatory. Over the next 3 days, make a daily observation of the animal's general health. One week after the injections, euthanize the mice with carbon dioxide and surgically remove the gel plug. Once retrieved, photograph the gel plugs alongside of a ruler. Then, Place each gel plug into a histological cassette and submerge them in 10% neutral buffered formalin overnight at room temperature. The next day, wash the 10% neutral buffered formalin away with distilled water. Store the cassettes in PBS at 4 degrees Celsius until they are evaluated. For histological evaluation, make 7-micron sections of the implant embedded in paraffin. Then, quantify the density of microvessels in the middle part of the implants with the aid of an H&E stain. Score the number of vessels per square millimeter. To demonstrate the human nature of the microvascular vessels, stain the sections with a commercially available anti-human CD31 antibody at a 1:100 dilution. This phase contrast image shows the characteristic cobblestone morphology of endothelial cells in a typical confluent monolayer of cord blood-derived ECFCs. This image shows the typical spindle-shaped morphology of human bone marrow-derived MSCs. After a week-long incubation in the mouse host, the implant, marked by a yellow dashed line, looked very different from the host's tissues. At higher magnification, multiple microvessels were revealed. They are luminal structures containing red blood cells and are highlighted by the yellow arrowheads. When the plugs were immunohistochemically examined, the lumens of the microvessels were reactive to a monoclonal antibody to human CD31. The cell nuclei were counterstained with hematoxylin.
Once mastered, this technology can be done within 1 hour if it is performed properly.